Hello, welcome to the Fiber Cell Systems mini webinar, Clinical Scale Production of Extracellular Vesicles in a Hollow Fiber Bioreactor. I'm John Cadwell. Extracellular vesicles are small lipid bilayer spheres secreted by most cells. They range in size from 60 to 150 nanometers and contain microRNA and various proteins. In some cases, they have demonstrated powerful regenerative capabilities. They are secreted in small quantities, making their production for clinical applications a challenge. A hollow fiber bioreactor is a cGMP-compatible closed cell culture system that has the potential to produce gram quantities of extracellular vesicles. The first attempts to culture cells outside of the body, or in vitro, was performed by Harrison in 1906. He was trying to culture frog neural explants onto plasma clots. We can see that a plasma clot is going to be a three-dimensional cell culture support. It took many years of the development of laminar flow hoods and proteolytic enzymes and the advent of plastic cell culture flasks, the two-dimensional non-porous cell culture support, for cell culture to become the ubiquitous technique that it is today. Anytime you want to scale up cell culture, it's all a question of just getting more surface area, more non-porous two-dimensional surface area into a space. All of these techniques, however, rely on culturing the cells as a subconfluent monolayer in the presence of fetal bovine serum, neither of which is a physiologic way to culture cells. All of these techniques, to a greater or lesser degree, subject the cells to what we call the feast or famine cycle. These are non-homeostatic, non-physiologic shifts in gas and nutrients. We first seed the cells into the flask, and they undergo what we call the lag phase. This is the period of time when the cells are attaching to the plastic, and they're also secreting their own cytokines to condition the media. Even in the presence of serum, we need to optimize the microenvironment for the cells. Then we have a period of time where we have the optimum cell culture conditions for correct cell physiology, and here we'll mention that it's just being indicated by the secretion of monoclonal antibody. And just when things are getting good, it's time to split the cells, because we must maintain them in subconfluent fashion and subject them to this cycle over and over again. A hollow fiber bioreactor is a fundamentally different and a more in vivo-like way of culturing cells. It's based on the principle that each one of these little fibers is like a filter, shaped like a drinking straw, about 200 microns in diameter, sealed in the end with a polyurethane phase plug so that anything that enters the end of the cartridge goes through the insides of the fiber, and typically we culture the cells on the outside of the fiber. We have media circulating to the insides, cells on the outside. We control this molecular weight cutoff of the fiber to be anywhere from 5,000 Daltons up to 0.1 microns. So we need to put that into perspective. A Dalton is the mass of a hydrogen atom. An amino acid has typically an average molecular weight of around 120 Daltons. So now we can look at a monoclonal antibody, an IgG1, which has a molecular weight of 147,000 Daltons. So we know it is comprised of about 1,200 amino acids. We need to put this in perspective to the size of an exosome. An exosome is typically 80 to 120 nanometers in diameter, so it's much larger than an antibody. So we recirculate media through the insides of the fiber, culture the cells on the outside of the fiber. This just shows how the cartridge is assembled. There's our potting material, fiber bundle. Side ports give us access to the interior of the cartridge. The area outside the fiber is called the extracapillary space. Loop of silicone tubing provides gas exchange with whatever gas is present in the CO2 incubator. We utilize a positive pressure displacement pump, not a peristaltic pump, to drive the media. There's two one-way check valves. We squeeze this tubing and we generate a flow rate of up to 160 to 180 mils a minute for recirculation rate. Hollow fiber bioreactors are fundamentally different from two-dimensional monolayer flask culture in three very important ways. The first is that hollow fiber bioreactors provide an extremely high surface area to volume ratio. Our C2011 cartridge has 4,000 square centimeters of surface area and a volume of 20 mils. This means we have 200 square centimeters of surface area per milliliter volume. So you can imagine a film that's 50 microns thick. This is the kind of surface area to volume ratio that we can provide, and it's how we can grow cells at such high densities. It's the only way I know of to culture cells at in vivo-like cell densities, 1 to 2 times 10 to the 8 cells per mil. It's this high cell density that allows the cells to create their own microenvironment and auto-support with their own cytokines. Secondly, the cells are bound to a porous support, not a non-porous plastic dish. What that means is that in regular flask culture, the cells are bound to a non-porous support and the nutrients come from the top down. When the cells pile up on top of one another, then bottom layer cells doesn't get nutrients, so they'll die and peel off and float away. In a hollow fiber bioreactor, the nutrients come from the bottom up. So this means that the cells bound to the fiber have the highest level of oxygenation and nutrients, but the cells are still free to grow in multiple layers in a post-confluent fashion. This means that passage number and splitting of the cells is not required. Lastly, the molecular weight cutoff of the fiber can be controlled and retains and concentrates secreted products. So we have a 5 kD and a 20 kD molecular weight cutoff. So keep in mind again that a Dalton is the mass of a hydrogen atom. So this should put things into perspective in terms of the size of the proteins that can be retained. So we retain proteins, antibodies, viruses, and most importantly, extracellular vesicles. Fiber cell systems, hollow fiber bioreactors utilize a positive pressure displacement pump, or we call it a squishy pump. We have 2 one-way check valves, generates the flow rate. Media goes through the insides of the fiber. Nutrients and waste products can cross the fiber. Secreted products are concentrated outside of the ECS, where we can harvest them simply by moving into a hood and using 2 syringes to harvest our secreted products. Silicone tubing for gas exchange means that it's a closed, biosafe system. The Derived pump is designed to fit inside any standard incubator. Gas is controlled by the incubator. Temperature is controlled by the incubator. It's a very simple system to operate. We utilize a thin cord for power to go through the door, so it's not necessary to use any plugs and waste CO2 in the back. The hollow fiber bioreactor module is designed to be easily removed from the Derived pump and moved into the laminar flow hood for aseptic manipulation. As Pasteur said, life does not spontaneously generate, so a good sterile technique is always a plus. Maintenance of the cartridge can take only 15 minutes per day. All we want to do is measure the glucose and harvest our product. Measurement of glucose is critical because it's the way that we monitor what's going on inside the cartridge. As long as the glucose uptake rate, or the amount of glucose consumed in a 24-hour period of time, is plateauing or increasing, then we know that the cells are happy and things are going well inside the cartridge. A glucose uptake rate of 1 gram per day empirically demonstrates that we have about 10 to the 9 cells inside the cartridge. Total amount of glucose in the media tells us when it's time to change the media. We simply unscrew the bottle and put on a fresh one. When the glucose is about 50% depleted, then it's time to change the media. We don't want to wait for the pH to shift. We want to keep the cells under their homeostatic conditions. There are many advantages to hollow fiber cell culture. First and foremost is that your product is much more concentrated. Because of the large number of cells and the small volume that they're secreting the product into, your product is going to be anywhere from 50 to 100 times more concentrated than what you get out of regular flask culture. 10 to the 9 cells represents somewhere between 20 and 40 roller bottles or 50 T175 flasks worth of cells. The cells are creating their own in vivo-like microenvironment, and we have demonstrated this results in uniform and complete post-translation modifications, and that this environment engenders a more in vivo-like cell culture production process. Because we're not splitting the cells, and the cells are not subject to any shear, we see a reduction in apoptosis. The cells are necrotic, but they don't blow open. And the presence of host cell proteins and intracellular DNA, as well as membrane fragments, is dramatically reduced when compared to flask culture. Because of the cells facilitating their own microenvironment, we have developed a product called cDMHD, which is our chemically defined, protein-free, animal component-free serum replacement. Cells at high density require less support from serum and or specialty medias. cDMHD has been developed specifically to take advantage of these different cell culture conditions found inside a hollow fiber bioreactor. And these conditions remain consistent for production over many months. I like to show this slide just as a reminder of the tremendous amount of cell culture plasticware that has to be discarded every time we split 10 to the 9th cells. With the hollow fiber bioreactor, we can maintain cells for many months of continuous production without passaging and without splitting. Exosomes are a subset of extracellular vesicles and have garnered a tremendous amount of interest in the research community in the past few years. This is primarily because exosomes secreted by certain types of cells, certain stem cells, and other cell types seem to have powerful regenerative effects. And we're just now learning how these might be applied in the clinic. So a viable way to culture large numbers of cells to produce large amounts of exosomes for clinical applications is a critical step in the advancement of regenerative medicine. The classical method for collection of exosomes from cells in flasks consists of culturing the cells in flasks in the presence of medium containing serum. And then when they're about 90% confluent, we'll remove the serum-containing media and replace with basal media without serum. This is to avoid contamination with exosomes that are present in serum. After 48 hours, the conditioned media is removed and the exosomes collected. We have found that simply substituting cDMHD for serum during this collection period can dramatically increase the production and secretion of exosomes by cells in these flasks. Hollow fiber bioreactors offer many advantages for exosome production. The first is that large numbers of cells can be cultured in a small space. Our C2018 cartridge that has 1.2 square meters of surface area can support in a range of 1 to 2 times 10 to the 10th cells. That's equal to about 200 to 400 roller bottles worth of cells, but yet they're secreting the product, secreting the extracellular vesicles into a volume of only 60 mils. So the secreted exosomes are concentrated. Depending on the cells, we have continuous production for several months of continuous exosome collection. If it is required to utilize serum to support your cells, we can put serum only in the circulating media. The factors that support cells will cross the 20 kD molecular weight cutoff hollow fiber filter so that we can put only basal media into the extracapillary space along with the cells. cDMHD is a completely cGMP compliant serum replacement and can be used for clinical grade exosome production. We're finding that mesenchymal stem cell proliferation may be limited. We'll say a little bit more about that in a minute, but other cell types, certainly any of the cancer cell lines, proliferate very well in our system. The other thing is that it's very easy to apply a defined stressor, such as heat shock or anoxia, consistently to large numbers of cells. Here we have one of our early direct comparisons between 130 T225 flasks and one of our C2011 bioreactors. Remember, that's our 4,000 square centimeter, 20 milliliter size hollow fiber bioreactor. Out of the flask, we got approximately 4 liters of harvest media, 9/10 of a milligram of protein total, 1.6 times 10 to the 9th total particles in this volume of 4 liters. One C2011 bioreactor, we have 4 harvests of 30 mils each, 28.45 milligrams of protein, 8.6 times 10 to the 11th, almost 10 to the 12th particles. So we can compare this to the flasks, and it's equal to about 500 T225 flasks in terms of the numbers of particles of EVs that we collected. Exosomes derived from human adipose-derived mesenchymal stem cells have demonstrated wound healing activity. This is just a quick example demonstrating the bioactivity of extracellular vesicles harvested from the bioreactor cartridge. Dosing was 2.5 times 10 to the 6th EVs per wound, and we can see the difference between our controls and where we've applied the exosomes. The production of exosomes in a hollow fiber bioreactor renders it cost-effective enough to utilize them in a commercial product. There is, in fact, this product on the market called Exophase that utilizes extracellular vesicles that are produced in a hollow fiber bioreactor. Each bottle contains 150 million particles. Actually, here they're called smarticles. And there are standardized cosmetic tests for demonstrating the efficacy of skin treatments. And we can see that we have significant activity for wrinkle reduction, skin luminescence, and cell renewal based upon collagen secretion. I have personal experience with this cream that does seem to facilitate the healing of wounds as well. This is some data that's been provided to us by one of our customers using our C2025D cartridge. This is a small cartridge with a surface area of about 450 square centimeters and a volume of about 3 mils. It'll support in a range of 1 to 2 times 10 to the 8 cells. The primary data to be derived from this slide is indicating that we have consistent and uniform production of exosomes by these cells, but we can see by the linear nature of their production that the cells themselves are not proliferating. We have seen this in several different examples of MSC culture in our hollow fiber bioreactor. I have a slightly different perspective on MSCs. Hollow fiber bioreactors are more in vivo-like. So we know that cells behave like they will more in vivo-like in a three-dimensional cell culture environment. So it's not surprising to me that MSCs are not proliferating in our system. One of the definitions of a mesenchymal stem cell is that they attach to plastic and proliferate. So the bad news is that we need a lot of cells to see the cartridge because the cells are not proliferating. Good news is we don't have to worry about the cells differentiating because if they're not proliferating, they're not differentiating. We have plenty of data that shows that the phenotype of the cells upon their seeding in the cartridge and after 30 to 45 days of culture in the cartridge are identical. So we know the cells are not differentiating under these conditions. Here we have some data that was sent to us by one of our collaborators in China. These are human umbilical cord mesenchymal stem cells seeded at 2 times 10 to the 8 cells total in one of our C2011 cartridges. They seem to have found a cell culture supplement that can induce the mesenchymal stem cells to proliferate. They added it on day 8. We can see that 200 milligrams per day is consistent with our empirically derived 1 times 10 to the 9 cells consume 1 gram of glucose per day. So we find on day 8 they add the supplement and the glucose uptake rate increases up to what would be consistent with 10 to the 9 cells inside the cartridge. These are primary cells, so we can see after about 45 days of culture, they kind of start to peter out. This is to be expected. This is some data from the same group, and they're going to be comparing the human umbilical cord mesenchymal stem cells cultured in 2D flasks with human umbilical cord mesenchymal stem cells and the EVs they produce in three-dimensional hollow fiber bioreactor culture. So the basic thing that we'll be looking at is going to be the flow cytometry data you see here at the bottom. Here we're just seeing a comparison of the flow cytometry data after 23 days of culture and after 55 days worth of culture. So the main thing we want to see here is that we're not seeing any difference really in phenotype of the EVs from day 23 and day 55. There's a further size comparison between the 2D produced exosomes and the 3D produced hollow fiber bioreactor exosomes. Slight change in size. We do sometimes see a change in protein to particle ratio, but the cell culture markers are also identical between the 2D and the 3D produced EVs. So the one C2011 cartridge is equal to about 225 T225 flasks in terms of EVs produced. So in summary, hollow fiber bioreactors are the most in vivo-like way of culturing cells. They are the method of choice for the culture of 10 to the 9th to 10 to the 11th cells in a single cartridge. We've calculated if you use 6 of our C2018 cartridges, in 30 days we can produce a gram quantity of exosomes in a volume of 10 liters. The concentration of the EVs is going to be 100 times higher than with conventional methods. We also see a reduction in membrane fragment contamination and apoptotic body contamination. It is the most in vivo method for culturing cells, and this quality of culture is reflected in exosomal activity. The closed hollow fiber bioreactor system from FiberCell Systems is compatible for cGMP production, particularly when coupled with our cGMP compliant CDMHD serum replacement. It also does permit the use of fetal bovine serum without endogenous extracellular vesicle contamination in the ECS. Overall, it's going to save you time, space, and purification costs, and reduce dramatically the barrier to entry for clinical applications of extracellular vesicles. So we can harken back to the first attempts to culture cells in vitro by Harrison on a three-dimensional plasma clot, and all the two-dimensional tools that have been generated since then. That three-dimensional cell culture in a hollow fiber bioreactor is the most evolved and in vivo-like way of culturing cells. Thanks so very much for your time. I hope you have an appreciation for how 3D cell culture in a hollow fiber bioreactor is a more in vivo-like way of culturing cells. If you'd like some more information on our placental co-cultivation and mesenchymal stem cell production from placental cultures, please have a look at our website, www.fibercellsystems.com, or have a look at the other webinar that we have on cell culture and co-cultivation. Thanks again for your time. I'm John Cadwell with Fiber Cell Systems.